Okay, great. Thanks for the introduction, Luke, and giving me an opportunity to speak today. My goal with this presentation today is to give a high-level introduction to the biogeochemistry configuration of the model and the historical simulations that we did during the V1 campaign. And since I recognize that many people in the project are not as familiar, and in the E3SM community are not as familiar with the biogeochemistry aspects of the model, I'm going to try and walk you through some of the top level results with some explanation about what's going on and why we did it that way. So the motivation why we're interested in general at all in looking in biogeochemistry, a lot of the motivation for this comes from the large uncertainty that exists in future changes in terrestrial and ocean carbon, and that obviously then controls the uptake of carbon from the atmosphere or exchange with the atmosphere, and that feeds back onto climate. And the changes in carbon varied really dramatically across different models in the CMIP5. The figure at the right here is showing the change in vegetation carbon among CMIP5 models, where in the historical period, you can see that the models are already pretty different from each other, but they become really dramatically different into the future. And you can't really see it in this figure, but it also turned out that the land models that included representation of nutrient limitations, especially nitrogen limitation, tended to have a weaker terrestrial carbon uptake than those that didn't include those representations. And this suggests that model structure and nutrient limits matter for the prediction of future climate. And so this is the topic that we focus on in our V1 simulations. And just to provide a little bit background and context for this work, there are essentially two different pathways by which carbon cycle and climate can feed back onto each other. One is the concentration carbon feedback. So this is where an increase in atmospheric CO2 concentrations directly causes a response of land and ocean ecosystems, which then in turn changes how much CO2 they take up. So especially this is due to the fertilization effect of increased CO2 concentrations causing more photosynthesis. So this is a dampening effect on climate. The second is the climate carbon feedback, where climate changes that result from CO2 increases impact land and ocean ecosystems, and those then again change the rate of CO2 uptake by those ecosystems. So this is mainly due to temperature and precipitation changes, and their impacts then on photosynthesis, plant respiration, soil respiration, and the abundance and distribution of vegetation. If you're in an area that's drought limited and climate change causes you to have more rain, you can grow more, for example. So in addition to, keeping these two pathways in mind, there's one more concept I want to introduce here, which is relevant to our simulation campaign. And that's something called the progressive nutrient limitation hypothesis. So we have these two mechanisms where CO2 and climate feed back on each other. And then layering on top of that, we also have nutrient limitations on ecosystem growth due to availability of other nutrients like nitrogen and phosphorus. And the progressive nutrient limitation hypothesis says that as you increase CO2, and so CO2 availability becomes less limiting, or other limits due to temperature or precipitation may be alleviated, then you, when those things are less limiting, then limitations from other nutrients like nitrogen and phosphorus become more limiting and reduce the response of ecosystems to increases in CO2. And so you would have a weaker feedback between CO2, the carbon cycle, and climate as a result of this progressive nutrient limitation hypothesis. And so our simulation plan addresses these issues, and through our V1 science question, which was, what are the effects of nitrogen and phosphorus on climate biogeochemistry interactions, and how sensitive are these interactions to model structural uncertainty, especially in how those nutrient limitations are represented? And so the model configuration that we use to look at this question is the E3SMv1.1 BGC model configuration. This extends the V1 water cycle configuration with a few bug fixes and turns on the biogeochemistry components in the land and the ocean. And the schematic here is a kind of high-level overview of some of the processes that are represented. In the land, we have two different approaches to soil biogeochemistry, which are called CTC and ECA, CTC being our default model, and ECA being an alternative that we use to explore structural uncertainty. Both of these include nitrogen and phosphorus limits on carbon uptake. In the ocean and sea ice, we also have biogeochemistry based on the biogeochemical elemental cycling model, which includes nitrogen, phosphorus, silicon, and iron, and also a representation of ocean ice biogeochemical interactions. And so with that background, I want to introduce our simulation plan. The design of the simulation plan that we use here is similar to and has some overlap with the C4MIP, which is the coupled carbon climate model intercomparison project. There are 4 Cs, I can never quite remember all 4 of them, but the C4MIP. And so we have, so the overall approach here is that we want to disentangle the response, the radiative response of CO2 through the greenhouse effect and the response of ecosystems to the direct input of CO2 to the ecosystem schemes, so the carbon fertilization effect. And so we do a series of simulations where we use different couplings of CO2 so that either the radiation scheme or the carbon cycle scheme sees either a constant CO2 or an increasing CO2. And so that's the general idea. The slides will be provided after the talk. I'm afraid I can't go into a lot of detail, but we can, by doing these different sets of couplings, we can disentangle those 2 effects. And so now I want to introduce some of our high-level results from the land model and land biogeochemistry. And so the first thing that we need to do before looking at the model responses to changes in CO2 is just to evaluate whether the model is doing a reasonably good job at simulating the carbon cycle and other, and also land physical properties against observational benchmarks. And in order to evaluate this, we used the ILAM global benchmarking tool, which is a tool that's developed by a team led out of Oak Ridge National Lab. And this tool evaluates land models across a suite of ecosystem and carbon cycle benchmarks, hydrology benchmarks, radiation and energy cycle benchmarks, and some forcing and relationships between different variables, benchmarks for those as well. And on this chart, which is also in our paper, the models are being compared against a subset of models from the CMIP5, including the CESM1 BGC, the third column from the left here, which is the predecessor model of ours. And then the mean of the CMIP5 models is shown third from the right. And then the 2 variants of the E3SM simulations are shown at the far right, the ECA and the CTC. And the way to read this chart is that on a relative scale, the more orange the color is, the worse that model is performing on that particular observational comparison, and the more blue the square is, the better it's performing. And so the overall impression I want you to come away with is that the E3SM in both of these land model configurations is performing better on these benchmarks than most of the CMIP5 models did, and similarly to the mean of the CMIP5 models. And for those who are really interested, we posted all of the interactive output from the ILAM benchmark online. And so I've included the link here in the slides, and after the talk, you'll be able to have access to that and go see those results and dig into the details. And so I just want to show a couple other results here. The first is the response of plant growth or gross primary productivity to the carbon fertilization effect. So here, the solid lines on this plot, these are time series of GPP over the course of our simulations. The solid lines are CTC, the dashed lines are ECA. And the red and blue lines are the simulations that include the carbon fertilization effect. So you can see the response of plant growth to increase CO2 in this these rising lines here, and that the CTC and ECA represent this similarly. If we look more in detail at the geographic distribution, we can see some patterns about how gross primary productivity responds to CO2 increases globally. This panel here is showing the GPP in the constant CO2 simulation. So this is kind of the baseline without increases in CO2, where it's highest in tropical forests, which is what we expect. And then in simulations where we have a physiological response, so the increasing CO2 to the ecosystems, we increase that growth in the tropical forests even further. But this is in contrast to the radiative response. So when we have a greenhouse effect of carbon, but we don't have the carbon fertilization effect, we see a pattern where GPP actually decreases somewhat in the tropics and increases at high latitudes. And this is likely because at high latitudes, temperature can be limiting. The ground can be frozen during parts of the year, for example. And so by warming those regions, you can increase plant growth in those regions. Whereas in the tropics, you may have regions that may be drought limited or sensitive to water availability. So then in the simulation with the full effect, where we have both the physiological and radiative responses, this is what the pattern that we get, which is not exactly a linear combination, but sort of similar to a linear combination of the other two. And in this case, we get a combined response that has increases in GPP almost everywhere in the globe. So then one other benchmark I want to look at from the land model, which is a really important one, is the total loss of carbon from land since 1850. And the main result here that I'll get to is that this is actually, we're doing pretty well against observations on this. But this is a really important metric because it controls basically how much carbon we actually end up exchanging between the land and atmosphere over this time period. And that's a really important control for the coupled carbon climate system. So a couple things I want to point out here. Again, the solid lines are our default model. The purple and orange showed that land carbon declines over the 20th century. This is mostly due to deforestation and to land use land cover change in general. Then in the blue and red lines, you see the combined effect of that land use land cover change plus biogeochemical coupling. And so in these simulations, you have increased plant growth because of carbon fertilization, which we saw earlier in the GPP plot. So this partly compensates for the loss of carbon due to deforestation, and you get this U-shaped curve. And this kind of shape of a curve where you have a loss into the mid-20th century and then an increase again in the later 20th century is supported by observations. And on top of this, I'm now overlaying some observationally derived estimates of the total loss in ecosystem carbon since 1850 from a paper by Kadewala et al. In 2013. So this is one of the benchmarks that we compare against in the paper. And it's a little bit approximate because I sort of freehanded this here. But what you can see is that we are in the default model in the fully coupled simulation, with the radiative and biogeophysical coupling, biogeochemical coupling in the red line, we're doing pretty well at getting close to being within this range. And I want to also just mention that there's sort of a caveat here that it's a little unclear where the appropriate year is for starting to count inventories of carbon. And so I think that may add a little bit of fuzziness to how most accurately to do this comparison, but we're pretty close to being within those ranges. And then the final thing I want to just briefly mention here is that this is one of the areas where we did see a difference, a structural difference in the behavior of the CTC and ECA simulations. And in this case, the ECA simulations, which are the dashed lines here, had a much greater degree of loss of carbon. And this was due primarily to differences in how the soil phosphorus was treated and how it was initialized in the two models that resulted in the ECA having much stronger phosphorus limitation in this set of simulations. So then with that, I want to switch gears to the ocean and sea ice and just show a couple high-level results from the ocean and sea ice. And so the first figure I want to show you is the net CO2 flux from the ocean to the atmosphere during the present day. And here I'm using the present day because we have an observational benchmark for the present day. So this is what we show in the paper. And so the red colors on these plots are fluxes of CO2 out of the ocean. So this is outgassing, and the blue colors are fluxes of CO2 into the ocean. So that's uptake of CO2 or sinks of CO2. And you can already see by these figures that we have some pretty significant biases in the model. So we have, for example, if you look at the Southern Ocean, significant outgassing of CO2 that's much stronger than our observational benchmark from the World Ocean Atlas. And when we looked at the total ocean carbon uptake since 1850, so a similar benchmark to the one I just showed for the land, the E3SM model is predicting only 93 petagrams carbon per year uptake on average since 1950, whereas the observationally derived inventories suggest it should be in the range of about 150 petagram carbon per year, and our model is significantly less than that and outside of all of the inventories we looked at and also CMIP5 models. And so this we think is a pretty serious issue. So I wanted to speak just really briefly to some of the potential reasons for that and to kind of provide some context for that, also to show one additional benchmark, which is the ocean surface chlorophyll. This is a proxy for the biomass in the ocean surface. And so again, at the left here, this is the model, and at the right, this is the satellite observational benchmark from the CWIF satellite over the recent present day period. And so we have, you can see just, we have significantly less biomass in the surface ocean. And this is maybe especially true around coasts and in the Arctic. So this is another place where those biases are really showing up clearly. So then just without getting into too much detail, I've got a list here of what we think some of the sources of these biases are. A really important one being that the V1 ocean model didn't have the parameterization of lateral mixing along isopycnal surfaces. So that causes a lot of problems for the PGC. I'll just leave it at that. Biases in the river and nutrient inputs, which are partly related to the changes in the model grid that needed to be compensated for. And possibly also some problems with biases in mixed layer depth. And then at the coasts, of course, there's also just partly a resolution issue. So most of these issues, the first couple of points in particular, are being addressed in V2 development, so we expect to do much better in V2. And so then I want to quickly show some results. Then that all of this was sort of focused on model evaluation. And then when I started the presentation, I was talking about the feedbacks between carbon cycle and climate that really were motivating our simulation design. And so now I want to come back to that and show you a couple high-level results from that feedback analysis. So in the carbon cycle climate feedback community, there's a kind of an established protocol for quantifying these feedbacks. And the two different feedbacks are quantified separately. So the first result I'm showing you here, this is what we call the beta feedback parameter, which is the response of the CO2 uptake by ecosystems to changes in the CO2 concentration. So this is the concentration climate feedback. And I'm showing a figure here from a paper by Peter Thornton in 2009, which showed the values of the beta parameter for a set of other models. And I'm just indicating with this blue square on top of there where our model sits with respect to those other estimates. It turned out that the ECA and CTC models actually gave quite similar values for the beta parameter, from 1 to 1.2 on this scale of petagrams carbon per ppm. And they're mostly lower than the CMIP5 models were in their values of beta, which is consistent with the progressive nutrient limitation hypothesis. There were some differences in the way that the E3SM modeled in our simulations. There were, excuse me, I'm stumbling over my words a bit, but there were some differences between the model simulations that were done for this 2009 paper and for our paper, which are summarized here at the left. And then in this next slide, I am showing the second feedback parameter, which is gamma. So gamma quantifies the response of carbon uptake to changes in climate, which we quantify by through changes in temperature. And again, I'm comparing against a suite of CMIP5 models. And the E3SM in the two configurations, again, had results that were relatively similar to each other as compared with other models, and in both cases were much weaker. So we had a much weaker gamma feedback parameter than most other models. So again, this is consistent with the hypothesis that models that represent nutrient limitations will have weaker responses to increasing CO2 in terms of their carbon cycle physiological response. So just then to summarize and give a couple outlook points, we conducted historical simulations with the E3SMv1.1 BGC model in two different land model configurations, both of which performed well on the ILAM land model observational benchmarks. There are structural differences. If you look in the paper, we describe these in more detail, especially in the nutrient limitation and partitioning of nutrients between different pools. But both configurations have comparatively, in spite of these structural differences, they have the similarity that they both have comparatively weak carbon climate feedbacks, which supports the nutrient limitation hypothesis and the hypothesis that these feedbacks are weaker in models where nutrient limitations are represented. In the ocean, we identified several sources of bias in the carbon cycle, which are being addressed for V2, and there are additional papers that have been published or are in progress that are going to describe various aspects of these simulations in more detail. And I also want to really emphasize again at the end, because I forgot to do so at the beginning, that this is the work of a really large team. I happen to be the person presenting it on behalf of the team, but many, many people contributed to this work, and some of those people are going to be leading these additional publications. So with that, I'm going to finish, and thank you all for your attention.